Okay, good morning. You're right here right now, so I could show you my professional dress, but this is it. So this would be something that I would wear to teach in. It's plenty long, not tight, all that good stuff when teaching children. So I'm going to teach you using Screencast-O-Matic. We used it in Sec 510, and I really enjoyed it, so I hope to bring it on to my future career and do little mini lessons like this for my students to watch at home if needed. So to begin, we'll go through like how a student would come in and prepare for my class. So they would come in at this purple door right here, and this right here is my classroom design. But this would be my table with daily supplies that the students would use. So on the table they would find like a pencil if they needed it and an eraser if they needed it. But they would also find their copy of these guided notes, so they're almost exactly the same as my PowerPoints, except we took out like keywords and lines and descriptors so that they would have to follow along and fill in. So they would pick up their copy of that. If the lesson took more than one day, they would just reuse the guided notes from the day before. They would also pick up their group work, which looks like this. So it'd be a single sheet of paper. And then they would pick up their independent work, which looks like this. Again, a single sheet of paper with some questions to answer. So that would be the setup. Also, and I use this a little bit towards the end with my middle school students, but I found it really important that I wasn't sitting at my desk when they came in, like right here. I was actually in the door to greet them, and I would greet them every day with a handshake. I am a smaller individual, so I'm about 5 foot, and because of that, I found that my middle school students really like to hug me, which is fine, but I don't think it's a professional thing to do. I don't mind hugs, but again, it's not professional, so I changed it. I taught them one day as a warm-up to shake hands professionally, and then every morning after that, I would have them shake my hand in the door. And that, one, prevented the hugging that was semi-inappropriate, and then two, also set them up. I told them that as they go into a job interview, their handshakes are super important. So that was one of the reasons why I did that, and that's how I would greet them every day. So they would all find their seats and their desks, the little red boxes, and then I would come up here, and the projector would be right here, and we would start the PowerPoint, which is right here, and it would already be up for them. Okay, so I hope that you can still see my face, but if not, I guess that's okay because I don't know how to fix it. But we would start, I would say, welcome to class. Today we're going to be talking about the cell growth cycle and reproduction of the cell, specifically mitosis, which we're going to learn more in depth later, but we're going to start with the normal cell cycle. So a bit of review. What is a cell? A cell is the smallest unit of life, so I would ask the class if they knew that, and just a general question, they would raise their hand. And then I would show them the pictures and ask which of these is made up of cells. So in a sense, I would be asking them which of these is living. So they would go through and they would take their guesses, and then they would be asked to write this in because this isn't in their guided notes. So the block of carbon right there is not living. So even though we're all made up of majority carbon or carbon-based life forms, carbon just in general, the element is not living. And then I would ask which of these are multicellular and which is single cellular, so they would go through and tell me. And I anticipate them getting mixed up on the midge, which is the fifth picture from the left of the screen if you're looking at it, because it does look like a single-celled organism, but it's actually multicellular. That's a mosquito larva. And then I'd go through and tell them what each one was. So this is the first one is a sundew. I would use a pointer. That's a carnivorous plant, so I would describe it a little bit. So the sticky pads right there, the insects get stuck on, and then the plant absorbs the nutrients that it needs from that. The next multicellular one is a Carolina anole. That one's very specific though, because if they didn't know, the anoles, when they get angry or frustrated or stressed out, they change from their bright green color to a brown color like that. So that's an adaptation of them. And then that is a mosquito larva, which is something we don't normally think about, but mosquitoes do have a larva form that looks nothing like their adult form. And then the single-celled organisms, the very first one is a yeast cell, and it's actually budding, so it's reproducing. And then the other one is a bacteria cell with a nice flagella tail that they use for motion. And then we move into the cell cycle. So this is actually the way that a cell lives its life. So the very first one, the interphase, is the big name for the G0 cycle, the G1, the S cycle, and the G2, and we'll go through what all of those means in a second. But we start with the gap 1 phase, GO, and during this phase, the cell is no longer dividing. It's not dead, but it's no longer dividing or growing, and the cell might have been programmed to do this. So some cells stop reproducing because it failed to meet a checkpoint. So on various parts of this cycle, there are checkpoints. So the cell stops and says, okay, do you have everything you need to move on to the next stage? And if it doesn't, it can get kicked into this gap 0 phase. And then some cells are just programmed to do this in general, like your cardiac cells and your nervous cells. They exit the cycle once they reach maturity. So after they have grown all that they need to, they stop and they don't go anymore. And I have a question about that. So I want you to apply what I just said about brain damage. So we know the danger, talking about football, of brain damage. So it's often irreversible. Learning what you know right now about the nerve cells and that they're programmed to just stop growing and reproducing, does it make a little bit more sense as to why brain damage is irreversible? And then the same goes with blindness, because whether you know it or not, your eye cells are actually nervous cells in the beginning. So when you damage your eyes or your nervous cells in your eyes, it's often irreversible. Yes, they can do laser surgery and like extensive surgeries to help your vision and corrective lenses and stuff, but a lot of the cells won't be able to fix themselves. They're not going to reproduce or grow. And then we move on. So just a general overview of the cell cycle. Each cell follows a repeating pattern of events. So the gap 0 phase is sort of out of the cell cycle. It can return, but it doesn't actually. So that's why we chose to define the cell cycle now. So each cell follows a repeating pattern of events that circulate in growth and reproduction. So this circle chart that you're seeing on the board is actually what a cell does again and again and again until it either dies or it enters the gap 0 phase where it stops. So there's 3 main parts. You have the interphase, which if you look at that very inner circle, it says it. So the interphase is the majority of the cell cycle. So if you look at a cell, more often than not, it will be an interphase. And then mitosis is where it's actively dividing. So you're dividing the nuclear components, your chromosomes, and then cytokinesis is where the cells physically divide. So they split from that bubble like this to 2 little circles. And then if we go into the gap 1 phase specifically, it's gap 1, not growth. It's often confused, but just get that straight in your mind. The cell will continue to go through normal growth. So, sorry, I'm watching the time. So in this stage, the cell organelles and components are duplicated, not the chromosomes, and then it just normally grows. It gets bigger. It gets ready to divide because you don't want to divide a very tiny cell. And then the synthesis phase is when the DNA is replicated, so your chromosomes are duplicated. So now you have, well, actually, memory check. If I finish the S phase, how many chromosomes do I have? So normally we have 48 chromosomes. So if I duplicate them, how many do I have? 92. Woo! So if you have 92 chromosomes, and that would be a question that I would ask the class, and most of these questions that I'm asking are ones that I would give them a second to answer, and if they don't, I would just continue on. But I give them a good amount of time in Dr. O's class. She talked about, and Dr. Allison talked about, or sorry, Dr. Allison Jones, there it is. She talked about the importance of giving the students a long enough time to where they can understand and process the question and to not answer your question super fast. Sorry, I'm going to escape and just do a little time check real quick. So we're at 9 minutes, so I'm going to keep going, do a couple more slides, and then I'll go through the guided practice. There we go. Okay, so if we go through the G2 phase, so this is gap 2. During this phase, the cell continues to grow in terms of cytoplasm. Like I said before, you don't want a cell to be very, very small. You actually want it to be big so that when you divide the cytoplasmic parts of the cells, you have enough to have a nice hearty cell to start the cycle over again. The cell prepares for division, so that can mean getting things separated, your parts of the cell that are only used during division might become more prevalent. And then it also checks the DNA for errors. So once we synthesize that DNA, we want to make sure that we've done it correctly, so it's correctly duplicated. So different proteins and organelles will go through and make sure that your DNA is exactly as it should be so that it can move on, because you don't want to create a cell that is not correctly. So you don't want to create a new cell that doesn't have the correct DNA, because it's not going to work well. Actually, that G2 phase right there, if we're talking about cancer, which I'll like to throw into my classes every now and then because I think it's a taboo thing that people just don't normally talk about. So I will try to incorporate it into my class whenever possible. Not in a morbid way, but just in a more information way. I think it makes it less scary and more of a real thing than this abstract thing that's just hurting their adults in their lives or children in their lives. I don't know. But in the terms of cancer, that G2 phase is often where the cancer cell will mutate those proteins or those processes or those checkpoints, and that's where we'll have cancer cells because the DNA is going to be changed into a way that's not good. So it's either going to code for the cell to divide, or it's going to turn off certain genes that prevent that the natural stopping of a cell from dividing out of control. So that's often where we'll have mutations. So then we go into the mitosis phase, which is not interphase anymore. So we've exited interphase and now we've entered mitosis. So it's the asexual division of one parent cell into two daughter cells that are genetically identical to each other and the parent. Excuse me. So it's asexual, which means that you only need one copy. 100% of the DNA in each daughter cell will be the same as long as the G2 phase did its job. And then cytokinesis, like I said before, is the final separation between the daughter cells. So at the end of mitosis, your cells are still connected a little bit. They're in the middle, but cytokinesis is actually the splitting of the cytoplasm or the movement of the cytoplasm. And then here's a video, but this is actually where I'm going to stop my mini lesson and show you just a little bit of what I would do with the guided practice. So we would go through the mitosis specifically, as you can see with all the slides. And then we would do our group work. So I would have them break into groups of 2 or 3, and we would discuss the 4 phases. So again, the group work looks like this. So they would discuss the 4 phases. They would fill this out for me. They would describe the events. They're more than welcome to use their guided notes. They're more than welcome to talk about it, and I would prefer it, and to not help each other draw the picture, because I'd like them all to draw the picture. But I'd make it very known that I'm not grading based on artistic abilities. I just want to see what they know. So that's what we would do. I would give them about 10 to 12 minutes for that, and then I ask them to return to their seats. We do another little review, so this is more of a game. The names don't pop up till later. And then we'd go through that cytokinesis, so the final splitting, talk about the differences between the animal cell and all that. And then we would do the independent worksheet, which again is this one. And they would turn that in, and I would grade them all. Okay, they'd get the remainder of class, which should be about 10 minutes to do that group sheet. And like I said, they can use their notes and all that good stuff. I hope you enjoyed my lesson. I'm sorry if any of it wasn't exactly as it should have been, but like I said, I really enjoy the Screencast-O-Matic, and I hope that you could see my picture through all of it, but if not, I didn't move. It just looked like this. Again, thank you, and I enjoyed the class, and have a great rest of the summer. Thanks.